Hello and welcome to the fourth part of our lecture on the scientific method. In this part, we will be discussing human interference, as human interference is an essential part of the scientific method, although philosophically speaking, the scientific method should be something truly objective. It's it's obvious that humans are the ones actually doing the science. Humans are actually the ones doing the scientific method, so they also interfere with the process of the scientific method. For instance, it starts already at the beginning when we start to define what are we going to research. This is obviously decided by the people that do the research, the scientists themselves. This can be a PhD that decides to study a certain phenomenon, or a postdoc or a professor that supervises research and tells the PhD student to investigate something. It can also be you guys, bachelor students in their thesis, or master students doing their final thesis work before they graduate. On top of that, there's also external sources or external people having an influence on what is research. For instance, the funding agency, as we will discuss in the next few slides. Funding agencies fund the research and therefore also have a say in what the researchers will do during their research. On top of that, funding agencies are often funded by governmental programs, and these funding agencies are therefore controlled themselves by the government and indirectly by taxpayers. So even non-scientists have a big influence on what happens in modern day science. The reason for that is that although we like to assume that science is truly objective and truly independent, grant writing and having people being paid from those grants is essential in science, because science, as we discussed in the first part of our lecture, is also a profession nowadays, and therefore the money has to come from somewhere, and grant writing and seeking funding is actually became a large part of the scientific method, the modern scientific method that is. And that obviously brings about a lot of problems with bias. You can be influenced subconsciously or consciously by the fact that you're funded by somebody else. You can also try to, what also happens a lot is that people have results and they try to make them a little bit more sexy, a little bit more appealing than necessary, just to increase the chance of getting publications, increase the chance of getting funding. Now, if you look at funding and how external sources influence research through funding, we have to know what type of funding routes there exist. Now, a very important funding route for us as scientists working at an academic institution is governmental funding. And this can be national funding, like we have the Nederlandse Organisatie voor Wetenschappelijk Onderzoek, or the NWO, in the Netherlands, that funds research at a national scale. You have the European Union that funds research such as Interreg, which is important for us, being the University of Maastricht laying in the border region between Germany, the Netherlands, and Belgium. For instance, in my group, we're doing research towards sensors that can help in food safety or medical diagnostics. And in this sense, we have two Interreg projects running where we focus on collaborative research, so research with a cross-border nature that involves both academic institutions and companies in the border region. Or you can do a Horizon 2020, which is a little bit more fundamental, doesn't require the participation of companies, but requires you to have multiple academic institutions or research organizations spread out to the European Union. So in this way, the European Union, with their message of cross-border collaboration, one European Union, actually implements that political agenda into science that is done in Europe by funding research. Likewise, the Dutch government also implements its political agenda into research by funding certain types of projects with certain types of goals. They direct research a little bit. And although many people think that is very dangerous, and it has some danger to it in the sense that researchers are not working truly independent anymore, it also gives you a little bit more direction as a researcher, and also uh, makes sure that scientists being paid by taxpayers' money are actually doing science to benefit society, not just doing science for the sake of science. addition to project funding, there's also personal funding. For instance, you can do a Marie Curie when you want to study abroad, when you want to do your PhD or a postdoc abroad. You can do an ERC grant or the NWO schemes Veni, Vidi, Vici, where Veni is for starting researchers. The difference here is that you also write a project, but the scheme is more open. So it's more research for research, but also aiming at high impact. So what they do is they judge your research project. They see, okay, like it doesn't really belong to a specific scheme, but it's very valuable for research and therefore indirectly for society. And they combine that with a review on the scientists themselves. So the scientist that writes the application, their resume, and their promise. Their potential for having a promising research career. So again, indirectly by funding them now, they get a tenure at a university, and they are going to do research for the next few decades that might indirectly, again, benefit society from this project. On top of that, sometimes the government, instead of making a little bit more broad, call specific topics, make projects where they just attract the scientists themselves. So it's not an open call that they have, but they actually make funding schemes or they assemble a group of scientists to work on a very specific project. Um, Examples of this are the Human Genome Project, where they deciphered the human genome, the Brain Initiative, where they work on neuropsychology, or the Manhattan Project during the 40s and 30s, where they developed the A bomb. They can also have funding agencies that are specified in funding research in one very specific domain, such as, for instance, a disease-directed funding, such as the Anthony van Leeuwenhoek calls that you have from the Dutch Cancer Institute that obviously always promote research that has something to do with solving cancer, diagnosing cancer, or therapy for cancer. And that becomes tricky. You also have private research funding. For instance, AXA has a research fund, IBM has a collaboration with MIT, and there's philanthropists such as Bill and Melinda Gates that also drive research, and that is a slippery slope somehow. On the one hand, it's very good because very valuable research comes from that and very professional research. On the other hand, it gives a little bit fuel, adds a little bit fuel to the fire for conspiracy theories because people from outside of science try to influence science. And although they do that uh, with the best intentions in mind, and although a lot of good research has come from this, it also endangers a little bit the position of being an independent researcher. And this is how funding being extremely important because science is now a very professional discipline can also lead to human interference. That can be good because it can provide direction and it can assure that the research actually has impact, but it can also influence scientists to do their job in a maybe not so objective way as they should. So it can also influence them to publish results in a certain direction that for instance, uh, suits IBM, just like giving an example here. So that is something you need to take in mind, but it doesn't necessarily mean that although there is interference, that it always leads to bias. When you think about the data part of the more complex, more realistic scientific methods that we discussed in the previous part of the lecture, you can also interpret the data in your own way without being biased. You do it objectively and come to a certain conclusion based on your specific expertise, and you discuss your data interpretation in your results and discussion section in your paper. Now, another scientist working in a similar field can have a different cultural background, a different scientific background, or even the same background, look at the same data, also in an objective manner, and come to a different interpretation. So the person that does the science in that particular case influences the outcome of the experiment, influences the conclusion of the experiment. Two different scientists can have a totally different opinion, totally different interpretation of what data actually means. And this is how human interference is not always equal to bias, but it also shows you that the part where we disseminate our research, our scientific communication, our peer review is very essential to do science in a proper way because a different interpretation can lead to fierce disagreement, but discussion can make you come to a consensus, can challenge you to look more critical at your data and come to a more thorough conclusion for your research. A way to overcome interference or a way to overcome interference leading to bias is something we discussed in the second tutorial we had last week, and that is reasoning. Logic is very important in interpretation of data, in interpretation of research, but also in the scientific method in general for making hypotheses, for studying literature. And there's two main forms of reasoning that are very important in the scientific method. One is called deduction, goes from a general fact to a specific fact, and the conclusion always follows from the premises with certainty. This means that the conclusion has no hidden information and all the information is in the premises. This means that if your premise is correct, your conclusion has to be correct. Let's give an example. You have a premise, only flowering plants have flowers. Grass have flowers. Therefore, grass is a flowering plant. So your conclusion flows from your premises with certainty. So there is only one option, and the conclusion can only be false if the premises are false. However, in science, also induction is very important. We go from very specific observations, we learn from those observations, and we come to a more general conclusion. Now, important difference here is that you can have premises that are totally correct and still come to a conclusion that is not correct. That is because the conclusion flows from the premise with probability, and the conclusion has information contained in it that's not necessarily present in the premise. For instance, all life observed until now contains DNA. So our definition for life is that all life contains DNA. We know that there are RNA viruses now that don't contain DNA, so we might have to refine our definition of life, but just based on all these observations and on these premise, you can come to that conclusion that all life contains DNA. And this shows probability. The more observations you have of life containing DNA, of living organisms containing DNA, the more likely your conclusion is thought to be true. Now, some philosophers of science, such as Popper, claim that induction, therefore, is not a very scientific way of thinking. Uh, Popper introduced the term falsification to demarcate between science and non-science, and therefore inferred that deductive reasoning is the predominant form, or should be the predominant form, of reasoning in the scientific method. And he said the only way of preventing interference, human interference, from hindering skepticism and leading to bias is to use falsification as a way to create a demarcation between science and non-science. He said a good hypothesis has to be falsifiable. This is what he calls falsification. Certain observations should lead to falsifications, for instance, in the black swan problem. A good hypothesis, according to Popper, would be all swans are white. So from induction, you could come to that hypothesis and make that hypothesis into a conclusion, right? You see a million swans, 1 million of the 1 million are white. So inductive reasoning would say all swans are white. However, if you encounter one black swan, deductive reasoning says that your hypothesis was wrong. And this is what Popper says leads to the ultimate truth, falsification, right? Because your hypothesis, that's where you started from. And although induction is useful to make hypotheses, it's not useful to come to truth. It's not useful to come to scientific knowledge. Therefore, the true scientific path of the scientific method actually lays in deductive reasoning to come up with hypotheses that can be rejected. And in this way, we learn to understand a complex scientific problem in a much better manner. That is also the difference between pseudoscience and non-science on the one hand and science on the other hand. In pseudoscience, for instance, homeopathy, we don't go looking for falsification. We have certain things we believe in, and because we believe in them, we want them to be true. So we make them non-falsifiable. For instance, in homeopathy, when somebody, a scientist, analyzes homeopathy and says there is no correlation between the homeopathic drug and actually getting better, a homeopath will say that no, that's not true. It's more about holistic view. You investigate it wrong, and it definitely works. Then again, when the scientist says homeopathy is too diluted to work. There's not even a single active substance in there. They create a different ad hoc hypothesis called water memory. So the difference between pseudoscience and non-science, for that matter, and science is, according to Popper, the willingness to let your hypotheses, your theories, be wrong. And that's the difference between science and non-science in the fact that that it leads to skepticism. And skepticism is, according to Popper, the most important characteristic for science. Thank you for your attention. See you in the last part of our lecture on the scientific methods.